Welcome to the Big Mighty Speaker Series, and welcome to Patrick Kroboth from U.S. Geological Survey. And Patrick, yeah, you have unmuted and videoed yourself, so it looks great. Thank you.
Thanks, Steve. Hopefully we can make it through this without any problems. Hi, everyone. I've been with USGS since 2017, so about for 4 years now. Before I started off, I wanted to explain the title a little bit. So the other invasive carp, black carp, just kind of in context with the other invasive carps, and I'm speaking about the Chinese aquaculture carps, which I'll go over in a minute here. But black carp are always kind of grouped in with 3 other species, and they're kind of that other, or the comma and when they're describing the species. But they're the unique species with potential impacts and concerns, and I hope you can learn some of that from this talk here. I wanted to start off, this is the image that Steve shared with the posting, and it's my title slide. It was provided to me by a commercial fisher in the Missouri River to report that he caught a fish to me, and I think it's a really good, he didn't realize it when he took it, but it's a really good representation of what we saw, or what I saw from reports in those first few years. Kind of initial low densities. The black carp is the one, and you should be able to see my cursor here as I point out stuff in my slides. But black carp's the one in the middle, kind of blue-gray. Stands out a little bit if you're familiar with fish, maybe not if you're not too familiar with fish, but initially low densities, kind of difficult to ID and kind of under the radar, but that's changing as time goes on. Okay, I wanted to start out with what is a carp? So we're all on the same page with species and reference. Traditionally, carps and minnows have been in the same family, Cyprinidae. There's been some recent taxonomic revisions that suggest a division in that group. It's one of the bigger families of fish, but I won't get into that because that'd be really confusing. I will say that for reference, a lot of the native minnows, so the Cyprinidae is the minnow family, usually refer to fish that would have the name minnow, shiner, chub. A lot of the local fish that you'd be thinking about tend to be a little bit smaller bodied. When you think about a carp or fish that share that common name of carp, they tend to be larger bodied fish, and there's several species that share that name. I have some of those listed here. The common carp, you're probably familiar with that species. Also, the species that black carp are commonly grouped with would be the 4 major Chinese aquaculture carp. You have bighead, grass, silver, and black, and I'll get into those in the next few slides. I didn't have a great slide to describe range, but of those Chinese aquaculture carps, they all pretty much range from East Asia. What the map I have here is actually from Nico et al. 2005. That's a biological risk assessment and synopsis for black carp. And that's a reference that I'll use quite a bit. It's a really good reference on the species. And if you want to follow up after the talk and learn more, I definitely recommend looking at that. The range of black carp is Eastern China and Russia, as you can see from that map. The Amur, the Yellow, the Yangtze, Pearl, and Red rivers are highlighted there in blue, and they're native to those large rivers in Eastern China. And the other species have a somewhat similar range. They don't necessarily overlap completely and have different systems that they occupy, but you get the general picture.
Hey, Patrick, this is Steve. Patrick,
yeah.
Can you hear me? Can I interrupt for a second?
I can. That's fine.
Awesome. Oh, everything's working good as far as I can tell. I forgot to let our audience know that if they have questions, we're going to get to the questions at the end of your presentation. And at the bottom of their screen should be a Q&A. There's also the chat. So people can put their questions in the chat, but the best place is in the Q&A function, and they can just print their questions right in there. I'll keep track of those, and at the end of the talk, I'll be sharing everyone's questions with you, Patrick. So I forgot to mention that in the beginning, so I wanted to hop in and let everyone know that. So sorry about that. Back to the talk.
That's okay. Thanks, Steve. I was worried we were having more technical difficulties. I'm glad it's nothing big. So I have here, common carp. You're probably familiar with the species. On the next few slides, I'm going to show you that list of species again. On the right-hand side, you'll see the USGS non-indigenous species database maps that represent the watershed units where they've been detected or reported. Those are all in maroon. So you can see here the common carp is pretty broad spread. Originally, they're Eurasian species. They were stocked broadly as a food fish in the 1800s. In Europe, they're actually a common recreational sport fish. And I encourage you to check out some of that. It's pretty interesting, some of the intensity that people get into with common carp, but less so in the US and North America. Next is the big-headed carp. They're planktivore, range throughout the Mississippi, Missouri River basins. You see some additional locations there that include various reports from other watersheds, not necessarily established, but reports that exist. Silver carp, another planktivore, range throughout the Mississippi, Missouri River basins. Again, additional locations beyond that range. This is a species you're probably most commonly familiar with, or you've observed jumping from the water. You're most likely to interact with these when you think of the carps. A lot of times walking on the banks of the Missouri River, you might find fish bones. I've commonly found silver carp operculum or gill bones. It's from this area here. It's a pretty dense bone. They tend to linger around. So probably the one you're most familiar with. One thing I wanted to note with this, it can be kind of confusing if you're not familiar with the species. All four are also called the Asian carp. Silver carp are their own species. Sometimes people refer to silver carp alone as Asian carp, or bighead and silver carp combined as Asian carp. So that can be really confusing. Those two, bighead and silver carp, are also sometimes called the big-headed carps. Jump too far ahead. Next is grass carp. They're herbivores, consume an array of aquatic vegetation. Their distribution has been facilitated by use as a biological control for vegetation. They've been stocked fairly broadly, a lot of times as a triploid fish. That's a functionally sterile fish. Triploids are produced, or triploidy is produced by the temperature or pressure shocking of eggs. This is an important one to note because they're the most morphologically similar species to black carp. They're commonly confused. So onto our species of topic, black carp, and I cheated a little bit. I used this slide as an outline as well. Black carp are molluscivore, commonly described as one. You'll notice from this map compared to the other maps that their range is a lot less than the other species. And I'll get into some of that information here. Just what I plan to cover, I'm going to go over the background on the species. I'll get into the distribution, general distribution, and some information specific for the Missouri River. I'll go over research that we've done at our lab, particularly in my group. Most of that pertains to wild captures, and that's done through the Illinois incentive program, or it's a bounty program run by the state of Illinois. Fishers that collect a black carp and submit it for biological sampling under certain criteria can get a $100 reward for that fish. We also have done some lab and field projects. Some of those are completed, some of those are ongoing. I'll go over that work. And then I wanted you to be able to leave here if you were interested in what I had to talk about, knowing where some resources were so you can keep up as things progress and learn some more in the future. So background, black carp are the largest of the Chinese aquaculture carps here. What I have pictured is the US record that we have reported. That was from Thebes, Illinois. That was 1,607 millimeters total length, or 63 inches. When I say total length, I'm referencing a measurement from the tip of the fish's snout to the tip of the caudal tail, so caudal fin. Sorry, I'm bouncing around here. I got a touchy mouse. So the two fin lobes pinch, so the tip of the caudal fin to the tip of the snout. That fish weighed 115 pounds, but you know, large fish aren't uncommon. The one that I just showed you in the upper right-hand corner was from the middle Mississippi River. The one in the lower right-hand corner is from the lower, the one upper right-hand corner is from the middle Miss. The one in the lower right was from the lower Miss near Mississippi. And just for reference, you can see, because you can never tell how tall someone is, but you can see that that's about the width of a standard tailgate. I think that's a Ford like F-150, just for perspective. I'll move this over here so I don't slide around slides again. So they're a large-bodied fish. They have a large mouth. A lot of times what a fish can eat is limited by its mouth gate. So large fish, large mouth, potentially pretty large diet items. And we've also found and seen in some prior literature that they have potential to consume a lot of of a certain diet item. So here is an example of some river snails. This particular fish that the sample came from, this is a subsample, had 69 of this relative size snail in its gut. And I'll go over some of our diet research in the next coming slides. So we've seen other taxa that have been consumed in pretty high numbers, chironomids, snails, in our work. One of the reasons they're concerned is because they possess these powerful teeth adapted to crushing mollusk prey. I've got a few pictures here. So we can do it with the cursor without bouncing around a bunch of slides. This is a picture of the pharyngeal teeth. They're not oriented correctly, but I have some here. And this is one of those sad things that if we were in the same room right now and it wasn't COVID, I could pass all these cool things around and you get to see them. But now you just have to see them on the camera that's hopefully working. And I'll hold them at a height that you can see. So these are the pharyngeal teeth. If you look at the image, hopefully you can see me. On the lower right-hand side, that's the orientation. I'm holding them in the proper orientation. This is the lower jaw. It's an adaptation of the last gill arch on the fish, and they're adapted for crushing mollusk prey. They have these molar form teeth. They come in a 5 and 4 orientations, so 5 on one side, 4 on the other teeth. These teeth are actually pretty interesting. They've been studied. You see the picture I have there from Hayat 2013. They've been studied for their strength as an engineering material. And these pictures that I have on the right are actually from Gidmark et al. 2015. So Nicholas Gidmark was a researcher who did his dissertation on black carp. They studied the functional morphology and feeding of the species. So you can see that jaw orientation, those jaws can move independently. Some of the neat things about them, I'll show you this too. This is the keratinous pad. So that's on the roof of the fish's mouth. This is the part on the roof of the mouth that's oriented like that. You can see this fish actually wore away that spot pretty well. Typically, this is convex, and here it's concave from where it's worn out so much. This was a pretty big fish. Another interesting thing, and this kind of looks like that image of the enamel, but this is one of the replacement cap teeth. You can see that in the picture to the upper right-hand side. And it's kind of cool. I've talked to my dentist about this before, but they basically, the way that their teeth adapt, and we've seen this in tank studies, is they can grow teeth better adapted for their diet as they feed. So as they're consuming harder food, their teeth adapt and strengthen to that. So you can see those teeth. They're down here in the musculature below the jaws, and then that pad is right here where he has PAD. So because of this adaptation, they're in, obviously mouth gape limitations, size of the fish, they have the potential to impact mollusk species, which should be their main prey, of which many in North America are already imperiled. So you're probably wondering how they get into the U.S. They were introduced in the 1970s and 80s for snail control and aquaculture. Those snails are an intermediate host for digenetic trematodes, and basically those trematodes or flukes affect fish stocks and aquaculture ponds. So the snails removes the threat to the culture stock. Most research that you'll find on the species has been for their uses of food fish in their native range, or for their uses in snail control and aquaculture. 2007, they were listed as an injurious species under the Lacey Act, which limited some of the interstate commerce of that species. Over time, there's been escapes with increased reporting in the wild. The first reported capture was documented in 2003, and that was from Alexander County, Illinois. I'm going to show you a map here in a second, but I wanted to highlight a few things first. This is the map that I'm going to show you is from the NAS website, the non-indigenous species website, and it's going to be a point map over time. So I think it shows a little bit better than what I have here, but you'll see that the first record was 1994, and that was reported escape from a flood event at an aquaculture pond in Missouri. But I will note that the species have been used in Arkansas, Mississippi, Louisiana throughout that time frame, and there just aren't reports. NICO, from their initial work, they found that in Louisiana, there were a lot of captures that probably went unreported in the early 90s from what the commercial fishers were telling them. You'll see a surge in 2015 with increased records from the incentive program, so the points will start getting a little bit more dense. And then also down south, and I'll show you that here, so that's around the state of Illinois. Down south in the Arkansas, Mississippi area, and in the Louisiana area, you'll see the points start getting more dense as well. And that's from outreach efforts by us to try and gather fish from select commercial fishers. We purchase fish from them to get baseline data for our various research studies on the species, since we knew that they've been down in that area for quite a while. So let's see if this works. Hopefully it does. I'm going to start us off at 2003 instead of 1994, just because it takes a while to get to 2003 from there. And you should be able to see those points fill in now. These are cumulative records. So you'll see the records over time as it fills in. One of the things to note, you'll see a little point over in West Virginia that pops up right there. That was an image from a pay pond fish identified by the state. No wild fish have been caught. So we've cycled back around, and you saw kind of the base there, basically the full map that I just showed you. Zooming into the Missouri River, I took a screenshot of that to show you. There's again that record from the report in '94. Most of the stuff we have comes from a sole fisher in the stretch of the Missouri River from the Mocaine, Missouri area down to Hermann, Missouri. So we don't have a lot of reporting from this system. They're likely present in other areas. It's just we don't have a lot of people that submit fish or report fish to us. So a lot of what we know comes from reports, really that incentive program or direct outreach. So records that we have come from commercial fishers, from biologists. You can see a Fish and Wildlife Service biologist there that collected one. And then in the past year, we started getting records from hook and line anglers. We've had bow fishers before, a few other sporadic collection methods, but we hadn't had hook and line captures until this past year. I don't know if that's an effect of the low stages. Most of those hook and line captures tend to be smaller fish and tributaries. One of the things that states have done to provide information is to submit this handout that you'll see here. It's the Keep Cool Call, what to do if you capture a black carp. Basically, if you catch a fish, retain that fish, euthanize it humanely, put it on ice, and give us a call. And that document should be updated here soon with some other contact information. And that'll be available on asiancarp.us, where most of the news and updates and outreach stuff is submitted through. One thing to note is that if you get a fish, please report it with a photograph of any suspected capture. And one of the things with this program that we've had trouble with, at least on the front end, when a fish goes to a new area and you get new outreach that occurs, is differentiation from natives. We've got these 4 species of natives that are kind of the most similar, or at least people mistake them and tend to report. That's the common carp, the blue sucker, and the grass carp. You'll see here I have black carp. These two up here are black carp images. This one is a black carp. The common carp is always distinguishable by a barbel at the corner of the mouth. So it's if you're not familiar with fish, a little fleshy barbel that hangs in the corner of the mouth. Common carp and grass carp, depending on the habitats that they occupy, the color of the fish can vary. They tend to be a little bit dark sometimes. I've worked in Florida before in tannin stained waters where fish just tend to get darker in general. That would be an example for grass carp where it may be darker. All these black carp that are pictured here are pretty much black, and you would expect that coloration from the name, but that's not necessarily true. A lot of the fish we get tend to be blue-gray or nearly black. Blue-gray is actually a pretty common color, and that leads to some confusion with blue suckers in the bottom right-hand corner here. But they're easily distinguished by the dorsal fin morphology. On a black carp, the dorsal fin is fairly short, a little tall, but it's fairly short. It doesn't run the length of the back. Like on a blue sucker, you've got that high section at the initial part, and then it has a shorter section of dorsal fin that runs pretty long down the length of the back. So those species, common carp and blue sucker, are pretty easy to tell apart, but grass carp can be pretty tough. So one of the things we've done, we worked with Robert Rabick with MDC and David Neely with the Tennessee Aquarium. We put together this key, and David did a really good job with all these drawings. They're phenomenal. I could never draw anything that well. no matter how much training or time I invested in it. So these keys should come out in the next iteration of the Fishes Missouri along with the other keys for that text. But our focus mostly with these fish has been the large juveniles and adults. We describe them as large juveniles because black carp mature at late ages in aquaculture, 6 to 7 years, and larger sizes. And I'll get into those sizes here in a minute, but we're talking about, 3 feet or a meter. They're pretty big fish. Prior keys had highlighted pharyngeal teeth and external color. I already told you the limitations with external color based on habitat. And pharyngeal teeth are difficult because it's pretty time intensive to extract those. You can see pictures of the pharyngeal teeth in the keys here and here. And I've showed you some of the molar form pharyngeal teeth for black carp. Grass carp teeth are serrated. Those teeth are embedded in some of the most dense muscle within the fish, and it takes a long time to extract them. They're very difficult to cut out. So we needed a method or a set of characters for quick external identification. I have 3 here. The first is the anterior lateral line angle. This is actually something from the species black carp's initial description that we hadn't seen since, but it's actually pretty good, other than the fact that scales can be lost throughout a fish's life and that can affect it. On a grass carp, if you're looking at that lateral line, and I have it highlighted here in red, hopefully you can see my cursor again. That red line is the lateral line as it comes off of the operculum or the gill plate. It continues in the pectoral fin area, runs, takes a decline, and then continues straight along the length of the body. On a black carp, it comes off that operculum, runs straight, nearly straight until it reaches the dorsal fin. It takes a dip and then runs straight down the length of the body the rest of the way. So that's a pretty good character there. Next, we looked at the lateral head morphology, and this came from a description by commercial fishers in Louisiana that black carp had a pointier head than grass carp. From that, we did a truss network analysis. We put some points on the images that we got in the lab of the fish that we dissected. Found that at the preoperculum, so this section of the head right here, a grass carp has a deeper head than a black carp, and a black carp from the eye to the preoperculum has a longer head. So black carp do have that pointier head, and grass carp have that deeper head. And next is the visible presence of the premaxilla or the upper jaw. That one's a little bit more difficult to show from the images that we use. So I have an image here from the Fish and Wildlife Service of the grass carp that's pretty clear. Basically, if you look at the fish from the head down with the mouth closed, that upper jaw on the grass carp, you see that break that I have highlighted with the red arrow. On a black carp, you don't really see that because they have a more subterminal or lower hanging mouth. We saw some plasticity in these characters, and we'd recommend if you're going to try and ID fish off of these that you use them in combination, because obviously you can lose scales and there's variation in fish and images. So moving on to our lab and field research, I guess I kind of have some of the wild capture stuff grouped into here. We highlighted some priorities just to kind of organize everything. But we have understanding life history. Under that, there's diet, age, growth, reproduction and development, and the species status in the wild. Next, we really lack any species-specific management tools, so we worked on trying to develop some of those. Also, detectability and distribution. I don't know if the stuff that I'm about to go over is collaborations with other researchers at universities that we work with, and then folks within my immediate group of coworkers and those at the lab at CERP. There's others working on genetics. Population genetics is being done by the Corps of Engineers in Vicksburg. There's diet modeling that's being done by NOAA, and some other various impact projects that are being done as well. So give you a rundown of the species life cycle. Again, Nico et al. Is the kind of the main reference for them. Reproduction is like the other Chinese cultured carps, and I'm going to use my cursor again to hopefully let you see what I'm talking about. Reproduction is this point 1, so the fish, again, moving upstream, migrating for spawning and flowing waters. Egg development and drift, so this is their picture of spawning. Egg development and drift occurs with downstream, and spawning likely occurs in late spring or early summer, and stimulated by a rise in water level and temperature. After spawning and development, the larvae, which I have those little arrows here, migrate into the backwater areas where the young and juveniles develop. And they also noted that, or suggested that some of the main stem was used for feeding and wintering habitat by subadults and adults. So that kind of moves on to the records that we have in the US. Young of year is a term we use for fish that spawned within the past year. So basically the really small juveniles. We have very little reporting or records relevant to larval fish or young a year. The ones that I have here are the two sites that we know of or that they've been collected. One is Dutchtown Ditch near Cape Girardeau, Missouri. There were records from 2015 until the past couple of years. Most of those collections were from Wes Sleeper with NBC. I believe he's discontinued since then, but he had pretty regular collections every year of fish in the upper image size class. And then the last one was from Gar Creek, Kentucky, and the Ohio River in 2018. That was from Matt Thomas from the state of Kentucky. I'm going to show you the spots here. So these are Dutchtown Ditch is an agricultural ditch near Cape Girardeau. Hopefully you can make out that vicinity map if you're kind of confused where we're at. It's southeast Missouri. This is an agricultural ditch. They've also collected fish from the diversion channel that's adjacent. And then Gar Creek, this is the lower section of the Ohio right before it meets the Mississippi River. And that is a floodplain tributary of the Ohio. Next, I'm going to talk about development. A lot of this is my coworker's research, Amy George. This is planned work. She's completed previous comparable research on grass carp, silver carp, and bighead carp, and we're trying to fill the knowledge gap here with black carp. And the goal will be to assess the developmental rates and other early life history parameters for black carp. Through this work, we found that the species is limited in culture, kind of due to that large size and late ages of maturation. They're also not produced or used very much anymore, despite presence in the wild. So we've been working to procure fish from the wild for broodstock, and pictured in the upper right here are some biologists with Louisiana who assisted with our attempts to procure broodstock. That commercial fishing community is kind of that dense red spot near Simmsport, Louisiana. They have had a history of working with black carp, collecting them, reporting them, and that was kind of the main spot when we were looking initially for black carp that we tried to get them. And they had the most confidence. And we got a few. We found throughout this process that it's very difficult to get the fish from the wild. They stress easily. We've increased or improved our handling protocols and our transport protocols, and we're working with some local fishers who are permitted to help us try to get these fish and try to build up the broodstock that we need to do this research, which would provide basically the data that goes into models I've listed here that simulate the movement and development of eggs based on physical and biological development. So it's kind of gives you the opportunity to get those parameters in a controlled lab setting for when you have something collected in the wild. Next is the status of the species in the wild. One of the questions that goes with that is where are the fish that are being collected coming from? This is led by a collaborator at Southern Illinois University in Carbondale, Greg Whitledge. Greg's work is focused on the use of the otoliths or the ear stones of the fish. The otoliths are nice because they provide, they're inside the fish, so they aren't reabsorbed like fin rays or anything else. They aren't lost like scales. They're internal and they deposit certain characteristics of the water chemistry. He's using stable isotopes that are captured in the otolith structure with laser ablation that he uses to measure the presence of those stable isotopes and compare them to different streams at different portions of life, so either origin or later on. The Fish and Wildlife Service, specifically Jennifer Bailey at Florida Cross Fish Health Center, is using a method of flow cytometry for ploidy analysis to determine if the fish that are collected are triploid or diploid. And both are finding a lot of wild origin fish. From Greg's conclusions, there's an expanding wild population supported by natural recruitment, but we are finding some introduced and escaped fish still present, either from the otoliths or we find triploids occasionally, very rarely. For this, CERC contributed age and gonad development data, and one of the main takeaways there is that based on the gonadosomatic index, or the GSI, the males are maturing probably around 800 millimeters total length, the females about 1 meter, but we've got some work to do there. We'd like to dig into that more with histology to determine the size, age, and timing of maturation. The problem with the gonad work is that, or reproduction work, is that there's some of the more sensitive tissues in the fish, and if they're not preserved properly, by that cool call method, if you don't catch it and put it on ice or it sits in the sun for a while, those tissues degrade and we can't use it for histology. Moving on to the diet work, this is kind of a point of reference for potential impacts. A lot of this is done by my coworker, Barry Polton. He's also in our river studies branch. Barry published a manuscript in July of 2019. The sample size for that was the initial 109 black carp that were collected from 2009 to 2017. Barry was finding that they had a pretty diverse diet from the samples that he looked at. Predominantly consumed benthic prey, native uniotid mussels and snails were present. Asian clams and zebra mussels were present, but in low percent incidence. So that's the question we always get with black carp is this invasive fish going to eat these invasive mollusks? And he found that they do, but it's just at a low percent incidence. So some of the other interesting taxa that he found were they were consuming mayflies. You can see some picture there in the lower right-hand corner, midges. Some of the midges were, quite a few midges were consumed compared to the size of the fish, and then also caddisflies. Barry also found that there's flexibility in feeding modes. There's an example here. I'm going to use my cursor again. These are subimago mayflies that he found, suggesting that they were feeding midwater column. There was other taxa that were consumed that he found were midwater column taxa. And again, that local consumption of numerous diet items of a single taxa. So I mentioned earlier the snails, chironomids, and things that were in pretty high numbers within individual diet samples. So the uniotid mussels specifically tend to be the main point of interest with a lot of this diet work. And ID has been difficult because of shell fragmentation. Our IDs are physical IDs, and they're always done by the MDC, Columbia, Missouri office folks, Steve McMurray and Scott Feynman. I'm always impressed with the IDs that they can get from what we give them, because I showed you the pharyngeal mill, the teeth of the fish, and they do a really good job of chewing up their food. And it seems like it'd be really difficult, but MDC does a good job of identifying these mussels. The bottom 6 on this table, from fragile paper shell down to purple lilliput, were those from Barry's initial manuscript. We've continued processing diet samples from 2018 through July 31st of 2019. We added 221 samples to that work, and we added 6 unionids to this list. And I'll note that other black carp consumed unionids, but at times they were unable to be identified because the shells were chewed up too much, or the identifying features like the cardinal teeth or hinge teeth weren't present that would allow for that identification. Of those 6 new species, the top 2, the hickory and the wartyback, were Missouri species of conservation concern. So with that same subset of the new samples, 21 new taxa were also added to Barry's list, 4 gastropods. I mentioned the 6 unionids, but 9 new bivalves total, 4 insect taxa and 4 other taxa. One of the interesting things that Barry's found is the presence of this fluke, the Spitogaster contricola, and Jennifer Bailey actually identified that for us. This fluke has direct development. So it is a mollusk parasite that spends its life in mollusks, but we found it within the diets of black carp samples. And at times it's the only thing that's present within that diet sample. It is an indicator of prior mollusk consumption. And from rerunning his numbers and looking at everything with consideration of this fluke as an indicator of prior mollusk consumption, Barry's found that the contribution of mollusks to the diet's almost doubled. So whereas they were kind of an opportunist, now they're kind of more in the realm of molluscivore with what he's finding. So I do need to note the limitations of our approach. So we're using these commercial captures, and as I've mentioned before, a lot of that is dependent on how the fish is taken care of after capture. And it's easy when you're a biologist and you're out in the field and you're running a net and you pull the fish in and you put it in your cooler full of ice. But oh, I bumped back a slide again. Sorry. Time of capture is important. A lot of times these fish are caught in hoop nets. The hoop nets might sit out for 24, 48 hour set. So that fish, depending on the time that it swims into the net, it has time to evacuate its gut. Also, there's that preservation after capture, as I mentioned. If it's not put on ice, the GI tract contents can continue to degrade while they're in the fish's stomach. Next, this is our bait development work. I mentioned the lack of management or control methods. We also lack capture methods at this time, but I'll discuss what we've been doing on that in the next slides after this. Basically, this is our approach to try to develop a control method. The goal here was to develop a single dose pesticide bait specific for black carp. There's 2 approved fisheries toxicants, Annamycin A and rotenone. And to give you an example, these are commonly used, or at least rotenone is commonly used. Annamycin is not commercially available at this time. Rotenone would be used for pond restoration projects. If you wanted to drain the pond and remove the existing fish, you would apply rotenone for that, for example. Annamycin A was chosen for this work because of its toxicity at low doses. It's more effective with less Annamycin A. And our anticipated application for this would be in the vicinity of endangered mussel beds. So if you have a mussel bed that you know exists and you have some indication that black carp are there, you want to protect that bed. It wouldn't be a broad spectrum approach, but basically be to protect that specific area, that specific bed. So the work we've done, and we've done quite a bit, but we haven't developed the single dose pesticide bait that we need at this point. We developed the delivery method. You can see that pictured here. Our attractant is a live mollusk. In this case, we've used Asian clams because they feed and they excrete and they represent the shape and size and search image of a diet item that a black carp would want to ingest. Our pesticide vessel is a glass ampule. It's a borosilicate glass tube that one of my coworkers, Ben Stallschmidt, has been able to bend and make and form into an ampule that we can put the Annamycin into to block hydrolysis, because if it gets in the water, as soon as it enters the water, it begins to degrade. So that's attached with epoxy to the exterior of an Asian clam shell. We've also done some forage size preference testing with Asian clams from about 10 millimeters maximum shell width to about 30 millimeters maximum shell width. And we found that black carp more commonly consumed the smaller Asian clams than the larger ones. Even though they attempted to ingest, they weren't able to break the bigger ones. And that's relative to a 550 millimeter fish. We've also assessed the ingested toxicity of Annamycin A. We did this in 2 carriers with gavage, ethanol, and corn oil, and we've done the completed baits. The completed baits was with ethanol and acetone as solvents for the Annamycin A. Annamycin A comes as a powder. Acetone is the common solvent in the commercial grade Annamycin A, and ethanol is also a suitable alternative. But both of those aren't very palatable, so we attempted that corn oil to see if we would have similar effects, maybe be able to get a more palatable pesticide that the fish would want to ingest. We found that Annamycin A isn't as effective as an ingested pesticide. It isn't effective as an ingested pesticide, but results are likely an effect of the contact time and middle ingestion of the pesticide based on that solvent palatability. And also all the experiments that we set up, we tried to replicate the environment that a black carp will be feeding in. So a flowing system, we have a high flow rate in our tank. We didn't want the ambient toxicity of a broken vial. Basically, the treatment's similar to the fishery management approach where you treat a water body. We didn't want that cloud of pesticide to be the thing that killed the fish. We wanted to ingest the pesticide and have minimal exterior effects. Also, this approach that we've been taking is to design a bait that should hopefully last a little bit longer, because when we started working on this, these fish were really low density in the field. Individuals may catch 20 fish a year, and that was a lot. So we wanted something that would last a little bit longer than some of the food-based baits. In the past, some rotenone-based baits have been designed with extruded feed-based pellet, but they absorb water and degrade pretty rapidly. Some of the limitations for this work right now going forward are we don't really know the distribution and potential target habitats of black carp because we don't know that much about them. So once we know some of that, we should be able to design some different baits that would help under those conditions. So under detectability and distribution, I wanted to note eDNA here. That is the process of detecting an organism through its shed genetic materials. I have a coworker, Kathy Richter, who's worked on some of that with black carp, and there's also been some work done at the Vicksburg Army Corps of Engineers lab, but I can't specifically comment on that because that's not my work. Just wanted you to know if that was something you were interested in, that some work has been done. As far as collection, I'll go through a review of capture methods. I'm going to go through our habitat use and movement research, which is mainly based on the telemetry study of the habitat use of black carp that is independent of the majority of the data we have, which are commercial captures. And then I'll go over collection efforts with commercial gears that relate to that. So capture methods. In 2019, we put out a review that was on the location and methods of reported captures. The background for this is in the species native range. They're commonly collected for early life stages. So basically like the larvae and smaller fish are collected and they're put in culture ponds for grow out, but there isn't a whole lot of information on that large juvenile or adult size. That's been kind of our focus and what we've gotten a lot of the information on the species from. We have a few descriptions though for this size. There's been baited hook and line catchers, drifted and anchored gill nets, trap nets, fyke nets, hoop nets. In North America, captures have occurred with hoop nets, gill nets, and trammel nets. And there was a prior study out of Mississippi that looked at the catch rates relative to fyke nets and alternated current electrofishing in culture ponds. So the goal for that review, habitats, time of year, and methods of commercial and scientific collections of biologist captures. The data were from 2011 to February 2019. We looked at fish dimensions and capture location. Data are dependent on commercial captures and effort though. So that's one thing I want to note is that capture location does not equal habitat selection or preference. So from that review, we had 302 samples. The size range was from 410 to, again, that biggest fish that we've seen. 50% of our samples were from 776 to 944 millimeters total length. You can see that from the red bars here, that's that middle point. The sources were mainly from commercial reports and we had a few from biologists and a few that we didn't have documentation from who reported or what source they were, if they were biologists or commercial fisher. So captures occurred by several gears, hoop nets and gill nets among channel, side channel, and backwater habitats throughout the year were the most common. Hoop net catch was highest in May, June, and July. And gill net catch was highest in November, January, and February. So basically we see this pattern, and this is the pattern that they report to us, that their commercial effort is in the warmer months, they're running hoop nets, a little bit easier to run. In the winter months, they're using gill nets, and those are either driven or set nets. So either they're driving the boat to scare the fish or push them into the net, or they're setting them overnight and checking them. And if you're not familiar with the gears, I had some pictures here and I was going to go over them really quickly. I forgot to mention one thing. The other habitats that we considered were tailwaters, the areas below dams, reservoirs, which are really just Kentucky and Barkley Lake, where we have in the Tennessee and Cumberland rivers, where we have records from, and then tributaries. But tributaries are limited for reporting because a lot of that is dependent on the locations where commercial fishers can set their nets. And a lot of states have restrictions on commercial fisheries within those tributaries. So one interesting thing about hook and line captures, and I had an image of a fish on the initial slide for this, that was a hook and line capture, is that they're not restricted by these tributaries. So that's an area where we can learn some more from. Gill nets are reported commonly, and they're very similar to the lesser reported trammel net. And I'll show you those here. So basically I mentioned a hoop net. You can see on the lower left, this is a hoop net. These are circular rings. They can be fiberglass, they can be metal frames. Number of hoops depends on the set. We run 7 hoop nets. This is a 5 hoop net. They have 2 throats in them. The design of those throats also impacts what fish you catch. You can see those here. So basically a fish swims in, it's set in current typically, or it can be set in without flow and baited. And the front will be tied off, but the fish swims into that current break. As it progresses down the net, it gets captured in what we call the cod end of the area where the fish is entrapped. So this is what we call a passive gear. The gill nets, and this is a tied down gill net. This is something we see a lot from commercial fishers. that they report. A regular gill net would be one where this is the float line, this is the lead line. It's set to the depth that you're fishing. On a tied down gill net, you have a little bit of extra depth, and you have lines that go from the float line to the lead line, like here, and they cut that depth so that you have bags within the net. And the fish with a gill net are being captured by their operculum or their gill flap. They swim in and they get stuck on a trammel net, which is another kind of net. They have larger mesh size webbing, and they're getting stuck in those pockets. Most of what we had initially reported were gill nets. They tend to be tied down gill nets. Trammel nets do get reported, but we don't see a whole lot of reports from trammel nets. So it is work to tease out tied down or driven and all that other stuff. It's not usually volunteered. So this is one of the things I wanted to show you that's changing. We've seen an increase in seasonal reports. This is the length, frequency, or the size data by count that I showed you on the last slide from that summary. The numbers on the left are a change. So in 2019, in September, one fisher had 22 fish out of 36 that were caught that were at 429 to 590 millimeters total length. And you can see that's a size that wasn't commonly reported in the past. And in the past, years before 2019, he'd typically get, a good year's 20 fish a year, and he was kind of averaging 20 fish a year. So he did that over that in a month in September 19. In 2020, in July and August, that same fisher reported 95 fish for those 2 months. So we see here, those are, again, fairly small fish, less than 600 millimeters total length. There was 23. 55 fish from 600 to 700 millimeters total length. I'm always kicking myself because I found a picture of the cooler when one of those big catches came in. That would have been great to use here. The fish on the bottom are from 2019, which are about the same size, so you get the point, but still not as good a picture. Basically, the takeaway I want you to see here is that many smaller fish have been collected in that late summer, early fall from this area. And this is actually the area near the confluence of the Mississippi and Missouri rivers. And that's been in the past 2 years. So things are changing, our understanding's changing a little bit. On to the next topic, our telemetry work. So really what we're trying to understand here is movement and habitat use, again, independent of that catchability. Everything we know now is from commercial fishers where they set nets, and where you can set a net is kind of dependent on the habitat. So this gives a unbiased approach to where the fish are. So our study area in general is kind of the middle Mississippi River below Price Lock and Dam and the lower Missouri River. But the majority of our data are collected by active telemetry, and the distribution of our effort is dependent on fish movement. I have much to report here currently. Oh, I forgot to mention that long-range movements are monitored with an existing receiver array. I'll go over the tags in a second so you know what VEMCO is, but that array is managed by MDC in the Middle Miss and the Upper Miss. There's other state collaborators in the Illinois, in the Ohio River, and then some of our USGS folks and MDC have receivers in the Missouri River that we're hoping they report relative to sturgeon research or other fish research. So again, don't have much to report currently. There's difficulties present with this work. As with everything with black carp, capture is difficult. We have similar post-tagging survival issues with capture. We've had the best luck getting fish in May and July. That prior slide, I mentioned those really high numbers of catches in that late summer period. The problem with that is the water temperatures are still really high. We went out at the end this year in September when the water temperature started to decline. We caught 2 fish, but it's the same thing. Those fish are kind of small. And that relates to what I'm going to go over on the next slide here is our tags. I want to show you our tags too. So we're using 3 tags in these fish, and they're fairly big. I don't have anything for reference other than my hand, but this is the acoustic tag. And can you see that? Hopefully. This is a VEMCO acoustic tag. It's a 69 kilohertz tag. It's about the size of a tube of chapstick. It's just a little bit wider. We also have a radio tag. This is a radio tag made by Low-Tech, and all these have a little magnet on it. That VEMCO tag had a magnet on it. That will be off if you found it. This one will have the magnet off if you find it. It has a very long 20 gauge radio antenna that will be sticking out of the side of the fish. And I'll show you on the next slide. Our contact information is on it. Hopefully you can see that. But it's got an address, it's got a phone number, and it's got an email if you ever find one. And then the last thing is the depth temperature logging tag. And we need to get these back if we can get any information. Again, my name, address, email address, and phone number are on there. If you find one, we would love for you to report it. That's something that we really need your help on, because if you catch a live fish or you find a carcass or one of these tags, please call or email us. Depending on where you find it, we may be able to learn something important. And I'll give you an example of something that we did here recently. We were able to detect one of these radio tags, and a coworker and I were able to locate it. It was in a dredge spoil pit, and this was from a cutterhead dredge. And if you've ever seen a cutterhead dredge, it's an intimidating piece of equipment. But speaking with the property owner and going over their operations, we were able to learn that the fish was tagged in July, and that either the tag or the carcass entered that spoil pit in probably around September. So we know how long that fish was at large, and we know where that fish died. If it dispersed somewhere up the Missouri River, for example, that would give us an idea of upstream movement and range. Also, it's good to know when your fish don't make it. So again, that antenna for the radio is a 20 gauge wire. This is how you can distinguish the fish. If you catch a live fish, hopefully you can see those two images. That's a surgery happening. That's the radio tag being inserted through the incision, and it sticks out of the side of the fish. Again, on the bottom here, you can see the radio antenna sticking down. If you do catch a fish, they should have a suture scar on the belly where that surgery occurred, and that radio antenna should be sticking out of the side. No matter what, if you catch a black carp, euthanize it. You're not allowed to keep it alive. Put it on ice, and please call your local resource agency or us, depending on where you catch it. And if it has the tag inside of it, the little tags are actually really difficult, and I'd be concerned if somebody tried to cut it out or to extract it from a fish to call us about, because I wouldn't want you to find just the big one and not the little one, which is the one that we really need to get back. So I'm going to start wrapping up this talk. Thank you for your time. One of the things that I wanted to note here before I begin that, though, is that this is not a presentation in a vacuum. We've all seen impacts from COVID, and us in particular, we found ways to continue to work under guidelines with precautions. For example, sterilizing surfaces, wearing masks, and wearing a long time. All different limitations. We follow guidance from the Department of Interior, the U.S. Geological Survey, our center, and the Center for Disease Control that established those guidelines for us to be allowed to work. I wanted to note that any photos that you see of staff without masks predate COVID restrictions and 2020. And there's an example here. This gentleman on the right was a commercial fisher who helped me back in 2017 when I was trying to figure some of this stuff out and get started. And we've had some of the gears that we use, and we've had to reduce our crews. So this is a 6-foot diameter hoop on a hoop net, and he's caught black carp for a very long time, and he knows quite a bit about catching them. And that's why I went down and met with him back then. And he suggested running these 6-foot hoops. We tried some of them at the start of some of our field work. And then with the coronavirus restrictions, we had to reduce our crews from 3 crew members down to 2. And it's very difficult to pull a hoop in that's that big with a big catch in it. So we went down to 5-foot diameter hoops. And we had luck. That's a fish that we caught, tagged, and have been following since last spring in that image there with the masked staff member. So I'm struggling with the pages. I wanted to provide some additional information to you, again, as a follow-up if you're interested going forward. I mentioned the non-indigenous aquatic species database several times, not just for black carp, but for any aquatic species. I really recommend this site, and it has a lot of options for you to look at and to follow. You can keep up on the distribution of species and learn about a little bit of their life history and various characteristics. AsianCarp.us is the main resource for posting outreach materials and information. On there are all some ID methods that I mentioned before. I know the Fish and Wildlife Service has a video for black carp ID. There's also some keys, and any news is going to be on that site. So if you want to keep up with that, watch that. And wanted to acknowledge our funding sources. A lot of our funding comes from the Great Lakes Restoration Initiative and the U.S. Geological Survey. Also, the Illinois Department of Natural Resources runs that bounty program that's provided a lot of the samples for the information that we have. I'll acknowledge my coworkers, Dwayne Chapman, my supervisor, Courtney Cox, Barry Polton, Amy George, and the other USGS and CERC researchers and technicians. I mentioned Greg Willage and Jennifer Bailey both collaborate with us on a lot of work. And Amy Benson runs the Black Carp page for the NIS. So she's the one that keeps everything updated. And I got to borrow a lot of the maps from that site because of all of her hard work. So we also couldn't do anything without the biologists from the following agencies who contributed all of our samples, and also the numerous commercial fishers who take the time to deal with me and ship samples to me and deal with FedEx and all that fun. So I want to thank them. And with that, I'll take any questions. I guess questions to Steve.
Awesome, Patrick. What a fantastic talk. You covered an absolute ton of stuff. And we have some pretty amazing questions, really, because Big Money Speaker Series crowd is a bunch of really smart cookies. So we'll try to hit as many of these as we can, but we'll see. Right now, I think we have 15 questions. So we'll just kind of see how it goes. First of all, Amy Meyer was wondering, are grass carp and black carp found in the same habitats in the wild?
So depends on the scale that you're talking about. Yeah, they're collected in riverine habitats and backwaters and in the main stem. And in our collection efforts, we've caught quite a few grass carp, trying to replicate some of those commercial fishers' methods, but we haven't caught many black carp. So if that's an effective density or the distribution, I don't really know. So the fine scale question, difficult to answer. The broad scale question, yes, they do kind of overlap with riverine habitats, similar requirements for life history and reproduction.
I'm going to ask you, Patrick, too, maybe stop your screen share.
Yeah.
That just might help the audio a little bit. All right, cool. Amy Meyer was also wondering, do black carp digest the shell material, or do they dispel it and only digest the soft organ material from mollusks and mussels?
So that's kind of a cool question. With the tank studies that we've done, we've actually seen that they can, so with our bait stuff and the forage size preference, we put cameras in the tanks with the fish. And we've seen that when they break a clam with their pharyngeal meal, they can actually reject some of those shell fragments. And then some of those fragments are taken into the GI tract. And usually you find the shell pieces, they're probably expelled, they're probably not digested. You can find them kind of in groups throughout that tract. They have an interesting GI tract where they don't really have like a break in the stomach and the rest of it. They just kind of have a wider part in the front, and then it continues down until the contents are evacuated. But yeah, so they can eject there, they can reject the shells on the front end, or they can just pass through the system.
Awesome. Steve Adams was wondering, and I'm going to mispronounce this, is antimycin A toxic to unionid mussels?
Not from the things that I've read, no. It's actually not toxic to catfish as much as it is to scaled fish. So one of its uses in aquaculture ponds has been, if you have a catfish pond, that you can treat that pond to remove other scaled fish that you might not want in that system without the same impact on the catfish. So no.
All right. Let's see. Jody Miles is wondering, what is the main predation of the carp / black carp in their native waters? And are they existing in as dense of a population in their native waters as they are in ours? So I think a broader question about some of the invasive carps in general, but also including black carp.
Yeah, so they're actually in pretty low abundance in their native waters. Some of that's impact from um, just environmental impacts. What was the beginning of that question? About
What's the
diet?
main predation of
Main diet.
carps or black carps? In other words, what feeds on them or keeps their population down in their native water?
Main predators? You know, I'm not sure. I'm not sure what the composition of those different species in their native range is.
Cool.
They have a probably pretty rapid growth though, kind of like some of the other species. So your native piscivore is probably the main predator until they exceed, again, even with predatory fish, when they exceed gape.
So great white shark basically is what you're saying. Just kidding. Sue Hamilton is wondering what method is used to humanely euthanize a carp?
For the size of those fish, it would be a sharp blow to the head is the approved method for euthanasia.
Okay. Aaron Delone.
There are other chemical methods.
I'm sorry, I missed that last part. I kind of walked on you.
Oh, I was going to say there's other chemical methods like MS222, but they're very large. So for the size that we see, it'd be better to do a sharp blow.
Sharp blow to the head. Gotcha. Your coworker Aaron asks, do species of Asian carps hybridize? And do you anticipate hybridization between black carp and the similar grass carp in North American rivers?
There's hybridization of bighead and silver carp. I'm not very familiar with that. And to answer the later part of the question, I don't know. It all would depend on timing and location of spawning. And if they'd be able to do that, I'm not 100% sure.
Right. Linda Vogt is wondering, why do you need to find brood stock? At one point you were talking about how you were trying to find brood stock so that you guys could actually breed black carp. And why are you looking for that?
Okay. So the difficulty with that is if you want to do early life history research, you have to spawn the fish. And we have at CERC, we have a biosecure facility where we can spawn the fish in the lab, indoors. The limitation, though, is that those fish aren't really available in culture. As I mentioned, they're available in the wild. So we need to get the broodstock fish basically to be able to spawn them to study that early life history component of the species.
Bill Muni is asking the $1 million question, are black carp edible?
Yes, and they're actually supposed to be one of the better tasting Asian carps. I've never had it before, but a colleague who was at the center, who actually recently went on to another position, went to China. Oh gosh, it was about 2 years ago. And I remember he was telling me of all the things that he tried, and he said that was the best one that he had. So, and I've heard that a couple of times. That's, again, their use as a food fish. So they should taste pretty good.
Have you eaten any, Patrick?
I haven't eaten black carp. I've had silver.
Cool. Okay, Jody Miles is also wondering if you know, like, does the Asian clam, which, you know, we now have escaped into Missouri, and the black carp overlap ranges in their native waters? Are you aware of that?
I don't, I believe so, but Asian clams are kind of difficult. There's a lot of, the specifics are difficult. I know even in America, there's some different subspecies and forms, and a more complex question or answer is tough, but yeah, they do overlap.
Okay. Paul Portaluzzi is wondering what native fish species here feed on mussels or other mollusks?
So that's a good question. Freshwater drum is kind of the main one. And if you look at their adaptation, if you're familiar with drum, they can get pretty big, but they wouldn't have the same gape as a black carp, and their pharyngeal teeth or pharyngeal mill, it's a little bit different. I don't have a picture available to show you, but it's actually kind of like a cluster of very small teeth. I'm not sure of the bite forces of those if you wanted to compare the two, but they're a little bit different. Researcher Greg Whitledge's student actually recently did a diet comparison based on a stable isotope analysis of carbon and nitrogen, looking at the level that the fish feed at, comparing black carp to freshwater drum and blue catfish. And he just finished up, he just graduated I don't think he's published it yet, but he's finished his thesis. And the black carp actually had a more broad trophic niche than the blue catfish and the freshwater drum. It's pretty interesting.
So basically, it seems like black carp in larger numbers might have a more dramatic impact on like a wider variety of mollusk species. Is that kind of what that seems to say?
Yeah, they would have different limitations. So again, they have that higher maximum growth, they have a bigger mouth gape, they have a pretty powerful mill. So even a thick shelled species, they'd be able to break, or a thicker shelled species, they'd be able to break. So they probably have some mussel species that may not be consumed by freshwater drum would be more likely to be ingested by black carp.
Cool. Paul was also
we
wondering.
definitely have room.
I'm sorry. Go ahead, Patrick.
I say we definitely have room for work on that. There isn't much diet research on what the fish can eat, what species. One of the things we're interested in is how shell morphology affects the black carp's ability to eat a mussel. There's different shapes and sizes to mussels. So like if you think about the Asian clam, they've got kind of a rounded shell versus some of our natives, which are kind of ovular, kind of thinner. So how that affects the black carp's ability to ingest and manipulate that diet item to be able to break it and consume it. It's like, so you picture like a native Uniana that's got that ovular shape. Can they ingest it? Are they just going to break off a piece of the shell? Are they going to crack it whole?
Paul Pornaluzzi was wondering if are commercial fishers targeting Asian carp, or are they bycatch? So especially these fishermen that you're working with, are they trying to catch some of these Asian carp species? Do they have a market for them?
Yeah, and there's actually a paper that came out and looked at commercial fishing effort throughout the Mississippi River Basin in the past couple of years. And traditionally, you've had markets that are that buffalo catfish market. I think in the past there were some common carp markets. They aren't as big now. Up north, though, kind of in this area, upper Mississippi River, you've seen increasing markets for Asian carp as some states provide incentives for harvest, either like an increased price per pound based on whatever you catch. They'll supplement that. If it's like, hypothetically, if it's 10 cents a pound, it may be like a 7-cent supplement per pound or something like that. Other places will hire commercial fishers specifically to target the species. So yeah, for Asian carp in general, there's an effort. As far as black carp, there's not really a particular effort. It's still kind of bycatch for other species.
Okay, we still have quite a few questions. How are you doing? You ready for more, or?
Good.
Okay.
Yeah.
Janet Moreland was thinking that those tags seem huge to put in fish. Like, why are these tags so big, and like, why can't we use microchips? Like, can't you just do like, I mean, like we all just got chipped with the vaccine. No, I'm just kidding.
I'll show you these tags again before I talk about them. So they are pretty big, and to be honest, when I was in grad school, these tags with a longer life used to be a little bit longer, about that long. The size of the tag, any tag, is dependent on the power output of the tag and the battery life of the tag. So ours are fairly short-lived. Relative to the size of the fish, though, this isn't really that big. These fish that we are looking to tag, and again, that's why I was talking about the fish from the end of summer, late summer, early fall, we're too small, because we don't want a tag that's going to exceed the mass of the tag is going to be over 2% of the fish's weight, is kind of a general rule. So you don't want a tag that's too big for the fish. That's why we tag a certain size fish. But there are limitations in technology. You know, we can't put a chip out there. You can actually, on the radio tag, if I hold it upright, you can see half of that tag through the epoxy battery, that purple and green part. And the rest of it is the tag part. The magnet is right there, but the rest of that, so about half of the size of that is the battery, just the power output. If you could do solar, so wildlife, for example, when they study different animals, they can put a solar component to that. Or some of the marine tags, the GPS tags for surface-oriented species, they can have a solar component to that recharges that battery. They don't need as big of a battery, and they just tend to be bigger because of the solar component. But some of the wildlife ones can be pretty small. They have micro tags now for that.
Oh yeah, that's super interesting. So this is my question. You know, when you're trying to study an invasive species like this, and you find these big fish, and so you tag them so that you can figure out what they're doing. In the meantime, they're like, you know, reproducing, right? Is there kind of a threshold where, like, as a scientist, you all make a decision like, you know, we're not going to kill every fish that we find. We're going to start tagging them.
Yeah, it depends on the level of where you're at with the study. So the area that we selected for this, there's quite a few fish that are caught. So there's established population that's there, and tagging a few fish is not going to be the same as if we went somewhere further upstream where there may be a barrier or something else present. There's obviously areas where we want to harvest or we want to keep the fish away from, like the Illinois River, for example. We wouldn't want to tag a fish up there. In the middle nest, we get a lot of captures. Most of our records come from this range. At this point, we need to learn something. It's that cost benefit of what are you learning versus what are you losing. So we need to learn something about where the fish are when they're not being caught, so that we can find a way to control them and have an impact on that population before it gets any worse. So that's kind of the stage that we're at. And you see that with a lot of other species as well. So another example I'm familiar with was like when I was younger, the state of Virginia did a tagging study on snakeheads, similar thing. So eradication of the species, but they needed to know what the exploitation or harvest rate was. The only way to get that estimate is to tag the fish to get reports back to understand how many are being taken out of the population by the methods that they're using. So there is a point there when you have an established population that you need to learn something to be able to control it better.
You know, Janet had asked that question about how large those tags are, and why can't you use something smaller? And your coworker Aaron Deloney chimed in with another piece of the puzzle too. He mentioned that microchips can usually only be detected a few inches to feet away from a microchip reader. Acoustic tags and radio tags, like those big ones shown, can be detected from hundreds of meters to kilometers away by the research boats. So when you're, you know, going through the Missouri River or the Mississippi, trying to find one fish that you've tagged, yeah, you know, it helps to be able to catch them from kilometers away. And I would have to imagine that that large power source has something to do with that, the distance that signal travels, huh?
Thanks, Aaron.
So Anne Egebrecht is asking, how do you dispose of carp? Can they be ground into fertilizer? So if you find them and you have to euthanize them, like, what happens? How do you get rid of them? And also commercial fishermen, if they find.
So there's a couple markets present for, you know, you talk about carp, Asian carp in general, or carp. There's a couple markets present for the different species. Obviously, food fish gets you kind of the highest price per pound. So if you're selling fish, that's what you're most interested in. There's protein markets. There you can use it for organic fertilizer. I've spoken to a commercial fisherman that did that up in Iowa. Yeah, it kind of depends on the number and effort that you want to put into your use. If you're looking, you have a few fish that jump in your boat when you're on the Missouri River and you want to put them in your garden, I'm sure that you could do that. The bones would be pretty big, as I mentioned. There's a lot of people looking into a lot of different uses for the species, and some different markets are available.
Cool. I guess that sort of makes me want to mention that we're, at the Wild and Scenic Film Festival that we're showing a week from Sunday. We do have a film called Trash to Treasure, and it's by a University of Missouri professor, Mark Morgan, who's been studying different markets for Asian carp. So basically trying to expand some of those markets and find markets that are available to commercial fishermen. There's a lot of work going on in that, but it's a cool film. I'm glad we're able to show that. Let's see, Gary Moreau is asking, he said, as a lake homeowner, we are told that grass carp are sterile. Is that true? And does that also apply to black carp?
That was what I mentioned earlier in the talk when I talked about the triploid fish. When they're functionally sterile, the eggs are fertilized and they're either temperature or pressure shocked after fertilization. So that leads to the retention of a chromosome. So they have 3 triploid fish. So they're functionally sterile to that point. Black carp, kind of a different question there. So that fish that's triploid is an aquaculture fish that underwent that process to be in that form. A wild fish is going to be diploid. It's going to be reproductive. You've got other questions with that too, though, of, within a system, is the fish able to reproduce based on the characteristics of the environment that it's in, if it's put into that. So that's a whole nother question altogether. As far as the black carp go, that process is actually a little bit more difficult than it is for grass carp. So some of the fish that are used in culture were used in culture were diploid because it was so much more difficult. As I mentioned, a lot of the work that we find, there's a large diploid wild population that's present. Most of the fish that we get are diploid. They're reproductively viable fish.
Awesome. And that really addresses the next question from Dave Erickson. And he was trying to get into that. What does triploid mean? So thanks for addressing that again.
Okay.
Steve Olson is wondering.
Like that.
Oh, I'm sorry. What?
I said 2 birds. That's not bad.
Love it. Yeah, we're getting there. We have 4 left, I think. Steve Olson was wondering, how have the East Asian mollusks evolved with black carp predation? That's a difficult question about Asian river ecology, really.
And I don't know the answer to that question, but it's a very good question, Steve. And I think that's something we can look into. I mentioned that there's a lot of work that can be done comparing the native range mollusks and diet items to what we see here in North America and impacts here. Again, that reference that I brought up several times, Nico et al., a lot of our diet work is based on their list of prior research within the species native range. And one of the things that Barry built his diet work off of was their list of taxa that they saw consumed. So that's something that we can look back at, kind of build on. So thanks, Steve. That's a good point. Something we can look into.
Another former USGS employee there. Chris Steffen is asking, how far upstream in the Missouri River do you suspect black carp occur?
I don't know. I'm not as familiar with the Missouri River because I kind of haven't had the ability to work with it. I haven't had the reports from there. I'm certain they go further up than O'Kane, Missouri, in that one record or that one Fisher's range. They have the ability to go probably until they meet a dam or a barrier that's present. There's actually been some discussion here recently. We were looking at taking that bounty program from the state of Illinois and seeing if we could do some increased outreach in the Missouri River Basin to try to get some records from there. and try to learn as much as we can.
Cool. So Brian Waldrop is, he works with the Missouri Stream Team program, and he's wondering, besides fishermen and fisherwomen and biologists, there's thousands of volunteers out on our big rivers throughout the years. What can these volunteers do to help further your research? Or are there any other ideas volunteers can do for you?
I think the things that I highlighted in the talk, those tags in particular, if you find one of those tags, and I know that's not an easy thing. Granted, the battery on the radio tag's purple, so that helps a little bit, and it's got white on it, but I know they don't stick out. I did get a call from a commercial fisher a while back that was looking for, I don't know, he was digging around on a gravel bar in the Middle Miss at some point. I think he found one of Aaron's tags actually from several years ago. So something like that would be a really big help to us. You can, I think there have been reports before based on people finding just the pharyngeal teeth or arch. Basically, things like that, outreach, just being familiar with the species. If somebody asks a question, a lot of times people that are out there are the ones that interact more than we do. We've only had this year with the low water was the first year I had a fish that jumped into somebody's boat without being caught. So it's not like silver carp where they're just jumping out of the water. I don't really expect that to happen, but you might interact with someone that catches one and wants to know what it is. Just knowing what it is would be a big help.
Awesome. I think this is the last question. This is from Ryan, who's wondering, do you think the insects found in black carp gut contents were bycatch while collecting mollusks, or do they seek out the insects? All those mayflies and caddisflies and everything.
Yeah, so that's a really cool question, and I didn't have a lot of time to dive into it, but it's a good question. So, gosh, I don't want to look it up because it's buried somewhere on my computer. But if you look at that manuscript that Barry put out, we've got some tables in there that are really interesting, and some of them relate to the midges, chironomids, and some of those fish that consume chironomids, they consume them in densities of the hundreds. So it's something that we think that they're actually eating them, even though they're a large-bodied fish. It's, this is a fish that's like 700 millimeters total length. We think that they're actually eating that food based on the densities that we're observing. So he's got some of that in there. I know he specifically has a table with the midges, and he has a table with the densities, the locations, so the general habitats. And another component that I didn't get a chance to mention from that work is that Barry also looked at the diversity of diet items consumed, the richness of diet items consumed among flowing water habitats and backwater habitats, or non-flowing water habitats. So in the non-flowing water habitats, there's actually a higher richness than in the riverine habitats, which is something also really interesting. So if you're interested in the diet, I'd recommend that paper. It's a good initial jump to look at that. And it's got more than I was able to go over here with the amount of time I figured I had to cover it and all the other stuff. So,
Patrick, is as far as you know, is Barry's research published through USGS? We could find it on the web about the diet.
yeah.
Okay, cool. Well, I'll find it and I'll link to it on the website.
You should have already linked to it when you went to our page at CERC. There's on that page for the project, there's a publications tab, and you should be able to access that publications tab in our diet paper and the other papers that we put out with capture and everything else if you want to read those. And as we go forward, we do more work, they should be added to that and available.
Well, that's awesome. Patrick, that is the last question, and thank you so much. At one point on Zoom, we had 77 people watching tonight, 20 more on YouTube, which is really cool. And even now, after 25 minutes of questions, I'm not sure what happened to my microphone. I don't know if this is still working, but there's still 50 people that are watching, and that's because this is so fascinating. We're really finding out about something that's new to a lot of us. thank you for the work that you put into sharing this with us. We really appreciate it.
Thank you. Thanks everybody for hanging in there. And I'm glad we got through the technical difficulties. I was worried a couple times.
Yeah, for sure. We had some glitches, but it worked. Great. Well, Patrick, have a good night. And to everybody watching, good night to you guys. And I hope that you can attend the Wild and Scenic Film Festival. Check that out on the Riverleaf website. And